Hello, everyone. Thank you for coming. All right, my name is Jacob Flores, and I'm currently a senior here at Houston Baptist University, and I'm a biochemistry molecular biology major. Last summer, I had the opportunity to go to Iowa City, specifically Iowa Carver College of Medicine, to participate in this program called Summer Undergraduate MSTP Research Summer Program, right? What a mouthful. But what this program is dedicated to doing is exposing students, undergraduates, in order to, or who are interested in pursuing an MD PhD, both MD and PhD. Now you might ask, what exactly is this dual degree program? Well, as the name implies, you'll be both getting your MD and your PhD. And there's a little split to it. So the first 2 years, you do your first 2 years of medical school, then you finish your PhD in about 4 to 5 years, hopefully, right? Then you finish out with 2 more years of medical school, and that gives you your fundamental knowledge to pursue medicine, be a practicing physician, or conduct research. The thing is, getting both of these degrees gives you a lot of flexibility. If you want to pursue lab work and that's all you want to do, you can do that. If you want to see patients and that's all you want to do, you can do that. And if you want a mix of both, work in a lab, be a PI, run a lab, and you want to see patients and learn from patients as well, obviously you can do that as well. Now, the program that I participated in, for 8 weeks, I was at Iowa City. And the majority of the time, I was, either conducting research under the mentorship, which I'll talk of a PI, which I'll talk about who and what I did later. B, I was also shadowing in either emergency medicine or shadowing other physician scientists. And there's also a lot of benefits. There's career development seminars that allowed me to know more about how to become an MD PhD and what is consisted of a physician scientist. And there's case-based learning, and there's just a lot of benefits that these programs have, especially this one. So in terms of the research, I was able to work in a vascular biology lab by my PI, Dr. Isabella Grumbach, and I worked closely with my main mentor, Dr. Karima Isa. So she was the one who led me through the experiments, talked to me project, and when I had questions, I would be the main one, but she would be the main one who I would go to. And the project that I worked on was dealing with how radiation therapy affects not cancer cells, but cardiovascular cells, specifically the cells that line the inside of your blood vessels, called endothelial cells. So the reason why this sort of project was thought up was, I guess the supporting material is from this case study. So what they did is that they followed children who survived cancer therapy, and they looked at them in terms of 10, 20, 30 years. And what they found is that the children who did survive cancer therapy had an increased development of cardiovascular disease, which can be denoted by these red bars. So the premise of the project was followed the idea that radiation therapy might have harmful effects on our endothelial cells, not just our cancer cells, so the cells that we want to keep healthy. So the idea was that irradiation is causing leakage in our mitochondria, which produces this ROS. What this ROS does, reactive oxygen species, it could cause deregulation of the cell, just causes stress, and just overall not a good thing for your cells. You can keep them from proliferating and functioning normally. So our hypothesis was that, okay, if we have a drug that's specific to that ROS that we saw here, and that can scavenge that, can remove that, can this reduce the damage from the cell? In this case, although I won't go too deep into the methods, we are looking at, I guess, cell damage or senescence. Senescence of cells is this sort of middle point where a limbo where you could either apoptosis and die off, or you can just stay alive and somehow bring yourself back. So we did a little bit of experiments. Of course, I'm not showing everything I did, but I could show you sort of the main pictures and the main idea that you can get from the effects of this drug named MTT, mitotemporal for long, MTT for short. Now you can see here, these cells, endothelial cells, were non-irradiated, so there was no radiation involved. These cells were irradiated, and these cells were irradiated and added with the drug. I can relate, or what one can relate is that the green and red colors that you see here is sort of like cell damage. These are proteins that are meant to like sort of keep the cell cycle under control. So as you can see, there's significantly less red and green here related back to the treatment cell category. So what's happening is that this MTT is actually having a positive effect. And we saw, there's another experimental design that we worked on in which sort of shows a similar effect. The more blue you see, the more cell damage, and the less blue you see means that there's a positive effect. The cells aren't as damaged. So there's a few conclusions that we could have made, that we made. One, therapeutic radiation is a critical influencer to pathological cardiovascular damage, specifically the damage seen in endothelial cells. Two, this mitochondrial ROS is detrimental to the endothelial cells, and the ROS scavenger specific to the mitochondria has observed to reduce damage. So that sort of was a big overview of the research that I conducted. However, there's a medical side, as denoted by the MD PhD route. And this actually was a really big turning point for me, because I did participate in a lot of research. However, I didn't know quite fundamentally if I really was interested in the medical side. So the turning point was this. I showed up for my first day of shadowing. The doctor, we went go and see the first patient. She says her symptoms, read her charts. We leave the door, close the door. He looks at me and says, "Okay, what was the most striking symptom that she had?" I didn't know. I just threw out anything I could. I said, "Okay, it was her inflammation." She was talking about her something on a chart, Crohn's disease, this and that, but all of them were wrong. And what it came to find out, it was while she was saying everything, she was coughing. So I missed that observation, and that really opened my eyes that although I could deal with little cells and like the protein pathways, all of that is great, but in the end, I want to work on research dedicated to serving the people. Medical-related research which intersects both the lab and patients, and serving patients. And I knew that was for me. And even further, I got to shadow a cardiologist, my PI, Dr. Grumbach. She also saw patients. And I could see myself doing the same thing she did. She was working in the lab, she ran the lab. My boss would say, "Hey, this is what I got," all the time. And when I finally got to see her as she went to patients and, telling her, like, not experiments, but did analysis and such as such as imaging of the heart, tests of the people. And I saw that she was able to implement both the ability to see patients and also work in the lab, and she was also an educator. And I could see myself doing the same thing. I saw that was probably the most perfect, ideal profession I could follow. And of course, it wasn't just shadowing and doing research. There's also workshops involved where I got to learn how MD PhDs became MD PhDs. I got to see how did they accept MD PhDs. I talked personally to the application committee people. I talked to students who are currently in the MSTP program, who are currently getting their PhD or finishing out their first 2 years of medical, of their first year of medical school. And I listened to a lot of innovative talks. As a matter of fact, this man right here was the director of the Bill Gates Foundation, one of them, named Dr. Jeffrey Murray. And I got to see how was funding done, and I just got to learn quite a bit as the program continued on. There's more than like 5, 6, 7, 8 talks that occurred. Now, of course, I wasn't just in the lab, just doing research or working on workshops, but I also had fun. I got to meet a lot of dedicated people, right, who helped me. who motivated me, other MD people who want to pursue MD PhDs. We motivated ourselves both in the gym, outside the gym, in the lab. So every afternoon we got off lab, we just went out and worked out. And I got to meet mentors who challenged me critically, who asked me why we're doing experiments, what we're trying to find, who coached me through as I work on this project. Of course, I show more opportunities that we had fun together, but more of the group. I got to see the Great Plains of Iowa, although it may not be much, right? Right? But it is peaceful. It grounded me. I felt, as I ran through this place, it was nice. And I kid you not, I ate the best ice cream I ever had in my existence, to this day. There was a jazz festival on the 4th of July. I went to a dive-in theater, just like the olden days. But when I really want to bring this together, there's 3 things that I can condense in terms of advice that helped me get to this point. 1, just talk to people, talk to strangers, get to know your professors. People here are just here to help us. 2, take advantage of education. I can guarantee that the education I got here at HBU helped me and translated to not only the lab, but in seeing other patients as well. There's times where I was coaching, teaching people just from like something I learned from molecular biology class with Dr. Whaley. And as you can see, I got to see the director of Bill Gates Foundation, which is also mentioned by Dr. Horn. And I also want to say, there's a lot of opportunities out there for you. You just got to explore and just apply. NSF and AAMC are government agencies or organizations that fund these summer research internships. There's tons and tons of these research internships. You just got to apply. You can go out of Texas. They'll pay for your flight. They'll pay for your dorm. There's a lot of opportunities. You just got to get them. And there's a lot of people who I want to acknowledge. The summer program for hosting me, of course, right? Dr. Chopik, as you can see here, right? Supporting me all the way through pies and not pies. She was the one who ignited my passion for research because I came in here just wanting to get an MD. But the moment I got my hands on research, I knew how to implement it somehow. Dr. Matsui, which wrote me the recommendation for this second internship, she was my PI last summer who helped me get into this one. The Grumbach Lag for hosting me, and College of Science and Engineering for allowing me to speak and for supporting me for these 4 years. And of course, my family and friends. And just as you can see, you can't do nothing without support. My mom came all the way to Iowa just to see me, and my dad as well, to see me succeed and reach my ambition. And this is the whole group as a whole. Thank you.
All right, so my name's Oscar Benitez Jr. I am a senior here at HBU. I'm a biology major with a minor in art, and also eventually I'll be doing a minor in Spanish, courtesy of Dr. Bermejo. And so today I want to talk to you guys about a life-changing experience that was very pivotal in the career choice that I've taken. I have a big passion for children. I've always been very fond of them, so I want to pursue a career in general pediatrics. So just the thought of being able to help out kids in the future is something that's driven me to kind of push forward. If some of you guys know me, I'm a non-traditional student, so I kind of don't fit the general criteria of someone that's pursuing such a long career path. But with that being said, today I'm here to tell you one of the experiences that actually helped me solidify and keep striving to reach that goal eventually, no matter how long it takes me. And so this summer I got to do a summer internship in Spain. So I got to intern during the month of July at the Centro Hospitalario Universitario de Santiago, or for short, CHUS. So I'll just say CHUS from now on, okay? So as you guys can see, by the little picture there, the little bunny, I was in the neonatal and the NICU, or neonatal intensive care unit of the pediatric section of the hospital. It is one of the top hospitals out in Spain, and so I was very privileged to be able to work there. So typically, my day consisted of internship for 4 hours every day, Monday through Friday, as well as going to a medical Spanish course at the University of Santiago's main campus as well. So for the first portion during the internship, I was able to do a few things like morning debriefs. So I was basically residents and doctors speaking about different cases about patients that were admitted to the hospital over the night before or over the weekend, if it was on a Monday. And so here are Lily and Monica. They're actually students at HBU that got to intern in other areas of the hospital. And my classmates were also Lily and Monica. And that's Profesora Rosana, which was an amazing professor. So like I said, back to the internship portion. I got to do patient rounds, so that was going and physically looking at every patient, checking their vitals, making sure if they were improving or not, if they could be moved from NICU to intermediate or actually be ready to go home. And for the course, it was just 1 hour, so there we had the opportunity to have guest speakers from different areas talk to us about the global healthcare system, but specifically about the healthcare system in Spain, which I was very astounded how very different it is to here. So you guys will find out how different it is when you get to see the stuff I got to do. So on the first picture on the top left was typical patient rounds. Besides that, I also had to, we were given a condition on neonatal patients. And so we had to actually give a presentation in front of the physicians. And if you guys have presented in front of your professors, wait until you present in front of a physician. That's very nerve-wracking. But it's also humbling too, because they're very welcoming, inviting. They gave me a lot of feedback, and it's a good way to expose yourself into the field. And this is the amazing crew that I got to work under. So from left to right, this is Doctora Ana, and the rest are residents. This is Cris, Loreto. Believe it or not, that's me without a beard. This is Blanca, and this is Doctor Alejandro. So those were the people that I got to see every single day, that I got to follow around. And they're very passionate about what they do. They're in the field of study that I want to pursue. And so getting to see them in action gave me an insight of what life would be like for me every day. The things that I did not expect to do, thanks to the openness of the healthcare system in Spain, is to actually get hands-on experience. So as you guys can see in the images, it's very immersed. It's a very immersed experience. So I learned how to do sterile technique before entering the operation room. And I actually got to assist in a few of the surgeries. So the one here is an orchidopexy. I will not go into detail about that, just for the privacy of the patient. But it's very hands-on, so we got to help out. I got to work along Doctora Marco, which was the surgeon that day. And then I got to observe an appendectomy, which is the removal of an appendix on a 2-year-old patient, so that was pretty cool. And it's just, it was very eye-opening for me because I'd never considered surgery as a venue in pediatrics, but that was amazing to see there myself. But even though that was fun and cool, the pivotal thing for me is the following picture. It may be a little graphic, but if you guys know me, I'm a very emotional person. I'm very caring. And one of my fears and one of the things that I prayed for when I took on this journey this summer was to see if I would have the capacity to endure seeing a child in such a critical condition. This child was admitted basically lifeless, if you guys notice. Her skin is very pale, so she has a lot of breathing tubes. She had assisted breathing. She had a lot of hormones being pumped into her system just to assist her with the very bare minimum to keep her alive. So to me, that was very difficult to see. And as you guys know the old saying, easier said than done. So I wanted to be exposed to that because I wouldn't want to pursue this career and then actually get there during my residency and either pass out or walk out of there because I cannot handle it. So to me, that was very important because I was actually able to be exposed and face my fear right before making the decision of either pursuing the medical field or change my route. And so the amazing thing is, after the 4 weeks that I was there, if the little clickers, is you guys can see the big difference. 4 weeks later, notice the pinkness in her toes. Maybe the light's not that good, but her skin tone changed. A lot of the assisted breathing and the IVs were removed from her. And so just a few days later, she actually got to go home to mom and dad. And to me, that is one of the most amazing things that I was able to experience or witness myself. I got to see how, you guys got to see that the majority of the physicians and doctors were females. And so they were able to give their everything and be passionate about what they do and still care, but draw the fine line about not putting their emotions in front to cloud their judgment. And they did everything they could to get that baby to go home just 4 weeks after being in such a critical condition. And so that to me was inspirational, and it gave me hope that I too can do that if I just push myself to continue forward. But besides the internship and everything, there was great experiences while going abroad. I've always wanted to travel since I was a kid. During my way to Spain, I got to go to Zurich, Germany. I spent the weekend in Portugal, just here. It's got some breathtaking views. This is Germany, and I think this is Zurich. And here, the Cathedral of Santiago was 5 minutes away from where I was staying. So that was pretty cool. It's the oldest cathedral in Europe, actually. So as a Catholic guy, that was very important for me to have within close proximity, so I had no excuses to not go to church. And you get to meet a lot of amazing people. Studying abroad pushes you beyond your limits. It pushes you out of your comfort zone. Even though I was a Spanish speaker, things are very different over there. The slang is different, so there would be some times where I was like, what? I have Spanish descent, so getting to touch base with my roots was amazing. This piece of bread is actually real. I didn't need it, though. And you get to build bonds with people. These are all HBU folks. These were all students that studied abroad in different sectors of the study abroad program, which Dr. Bermejo introduced me to. On the bottom right were different students that live with the host family. And here on the left is Maria Antonieta and her son, Nicolas. And if you get the choice to study abroad and they offer you the choice between a dorm and living at home, please live at home. Like, it's very different, especially if you live on the dorms like me for 3 years. It's not fun. But they make you feel like you're home and you feel like you're part of their family. So all of these things were very life-changing for me. And one of the ways that this was made possible, this was obviously an expensive trip, was thanks to the Gilman Scholarship. The Gilman Scholarship is actually available to anyone that's a Pell Grant recipient. So if any of you guys in this room are considering studying abroad or just venturing out, this scholarship gives you up to $5,000 actually if you are one of the selected individuals. These are the stats from last year, but I was able to get my hands on this year's stats. And actually this year there were less recipients. There were only 1,424 recipients. And out of those, myself and another colleague that goes to HBU were recipients for the scholarship this year. So even though the odds might be against you, just don't be afraid and try. Face failure, accept it even if it happens. If you don't figure it out the first time, do it again. It doesn't matter. Like I can't stress enough like how many times I've failed on my journey here. And what I can tell you guys, especially as undergrads, is don't let that stop you. Don't let that be your limit. I want this so bad that I'm willing to retake classes if I have to. I want it so bad that I will take summer courses if I need to. So you need to ask yourself that question and say, if this is the route that you want, how far are you willing to go for it? So that's my invitation to you guys. And in closing, when God gives you something you prayed for, never forget to give thanks. Above all, God was the reason that I was able to experience this experience, because without Him, I wouldn't have it. And of course, He gives you people that will help you get there. That being said, I'd like to acknowledge Dr. Chopik. She's been the person that's believed in me from the very beginning, even when I didn't. So thank you so much for that. I'd like to thank Dr. Bermejo for introducing me to this summer program opportunity, my family, my friends, the College of Science and Engineering, the people at the hospital, as well as the staff from the University of Santiago, the Gilman Scholarship, and my dad, even though he's not here, he's a very important person of me, and I carry him with me. So make sure that you guys have a foundation of support system. That's very important to help you make your way. This is a very lonely path, whether it's the MD or the PhD path. It's a very lonely one, but only if you let it be. So thank you so much. Put that like on your side. And then
Hi, everyone. Can you hear me? That's fine. My name is Peter Boland, and I'm a senior here at Houston Baptist University. And I am majoring in biochemistry, among other things, including a psychology minor. And I wanted to share with you all my summer research experience at Baylor College of Medicine and Dr. Ramiro Salas' lab in neuroimaging. So Dr. Salas' lab is interested in studying depression, anxiety, and drug addiction. And for my project, I'll be focusing mainly on depression side of things. As for the software that we use, at least for my project, what I used in my lab, we use other things in the lab as well. I use FreeSurfer for processing the anatomical information from magnetic resonance imaging. And from FreeSurfer, we can export that data onto Excel sheets. I also use the statistical package for social sciences, or SPSS, to create some figures and also to analyze some of the data. So psychiatry and using neuroimaging it's in its infancy. It's not really something that we use. When you go to a psychiatry clinic, usually they'll just assess the patient based off of behavioral data, but if we could also categorize psychiatric disorders by neuroimaging biomarkers, that could be helpful as well. So following that line of thought, my project is investigating whether changes in structural neuroanatomy of patients correlate with clinical outcomes. And one of the ways we can measure depression severity is through the Patient Health Questionnaire 9. For my patients, they had to have at least moderate depression, so scores of 10 or more. And the ages was 28 males and 25 females. The gender was 28 males and 25 females, and the ages averaged to 27 years old. And their length of stay at the Menninger Clinic was 55 days. As well, I have some preliminary results that I would like to share with you all that I found through an exploratory analysis that to give you an idea, the patients had to go in, we have about technically 700 patients. 53 is a small number, of course, but not all of those 700 patients had admission scans and discharge scans, to clarify. And in case you don't know, the Menninger Clinic is a psychiatric clinic in the Houston area near Baylor. I feel that I should also mention to you how I found, or how did I really do the exploratory analysis. For the before and after scans, I use a paired t-test to see which regions differed significantly under a 95% confidence interval. So for my project, I found, at least for one of the things I found, was that the superior frontal cortical volume correlated strongly with changes in the depression severity scores. With this figure that I made through SPSS, okay, I would like to orient you on the graph, which here you see on the x-axis, the changes in depression severity scores. So you see -30, -20, and so forth. The patients on the left towards the origin, those are having a lesser severity of depression. In other words, they're getting better, if you want to say it that way. And on the y-axis, you have changes in the superior frontal cortical volume. So you see 2,000 at the top in millimeters cubed. In other words, they had a larger volume in their superior frontal cortical volume. And here you also see a negative correlation here. So as depression severity decreased, superior frontal cortical volume also increased here, the Pearson's R value was -3, -0.3, and the p-value for that was 0.025, which is statistically significant with a 95% confidence level. As for some of the limitations, well, if we could have perhaps a larger patient sample size, so since 53 is really small compared to, say, 700, if we could have that data, unfortunately, but some of you may be thinking, well, what does the superior frontal area do? Well, so that superior frontal area deals with higher order thinking. In terms of treatment methods, we have some medication data. Not all of the patients were using antidepressants. And a further thing that needs to be investigated is to ask the coordinators at the Menninger more about how they're treating the patients there. I could also talk about the transcranial magnetic stimulation part, since that was interesting to me, since some patients who want to be treated for PTSD or major depressive disorder also receive transcranial magnetic stimulation, or simply TMS, I'll just say TMS, on the prefrontal area. I have to confirm what they're doing with the patients there, at least. And I want to thank my current PI, Dr. Ramiro Salas, and my first mentor at Baylor College of Medicine. He retired from Baylor recently in May. And Dr. Savannah Gosnell, who finished her PhD in Dr. Salas's lab in May as well. And I would also like to thank Dr. O'Hara and Dr. Chopik for their networking efforts, and also Dr. Wingate, too, my advisor, and also the COSI faculty administration. Initially, I'm happy to answer questions as well. The plan is to answer questions afterwards. Okay.
Hi everybody, my name is Arshina Rahman. I'm a senior here at HBU, a biology major, and I'm here to present a little of my summer research to you guys. Okay, there we go. So this summer, I had the opportunity to do research at McGovern Medical School here in Houston in Dr. John O'Brien's lab, and we were studying retinal regeneration. So as of 2010, the World Health Organization estimates that about 246 million people are visually impaired. Of this 246 million people, 33 million people are blind. And it's important to realize that the cost for vision loss per year from 2010 was $3 trillion, and this could have gone up. So basically, diseases of the eye and vision impairment are a burden socially, economically, and physically. So currently, there are only a few treatments for retinal degenerative diseases such as age-related macular degeneration, and another disease called retinitis pigmentosa is another disease which causes retinal degeneration, and the prognosis for these diseases usually end up with people being blind, or at least legally blind. But it's important to realize that if you were a zebrafish and your retina was damaged, it would be able to repair itself and your vision would be restored in a couple of weeks. So the problem is that mammals are unable to regenerate their retinal neurons that are lost by damage and disease. These zebrafish are model organisms to study because retinal damage has a regenerative response that can replace rods and cones. Oh, no. No. Oops. Sorry. No, you're good. Might be easier to use that. Okay. So sorry about that. So what is regeneration? Regeneration is the ability for the body to replace damaged body parts and fix them. So by identifying the signals that are triggering the self-repair response, we were trying to see if we could implement these into human retinas. So our basic testing method looked like this. This is just a basic breakdown, and we used a P23H mutant model. This is just a zebrafish that has a photoreceptor degeneration similar to humans, and we would sacrifice their retinas. And this summer, I received animal training, and we learned how to humanely do this and the ways to accordingly do this, and then we would gather data. So we would use immunohistochemistry, we would stain, and we would look at these retinas under confocal microscopy, and right now they're actually analyzing the data that we got from all this testing. So I want to show you guys a beautiful picture of how regeneration occurs in the zebrafish. This is a 33-day long regeneration. And so in the green, we see cones, and then in the blue, we see other kinds of cells. But in the latter pictures, in red, we see regenerating cells. So what we see is that the cones, as they're dying off, regenerating cells are able to come in and replace these cones. So this process is unique to zebrafish, and that's what we were studying, because it's interesting to see how this happens and how it's signaling and how we can relate this back to humans. So overall, I really enjoyed my summer research experience. I was able to learn new lab techniques and meet new people, especially get plenty of advice from new mentors. And it was really great to experience a higher level lab rather than an undergraduate lab and learn new things in general. So I would highly recommend that if you're a freshman, a sophomore, even a junior, to go try it out and network and try to get yourself into a research experience, because you'll never know if you'll like it. And lastly, I'd like to thank the HBU Science Department as well as Dr. John O'Brien for letting me do research in his lab and learn new things. Thank you.
Just get this wires. All right. Clicker. Hello, can you hear me? Yes? Okay. Hello, my name is Tristan. I did research in the medical center this summer at Baylor College of Medicine with the SMART program. It is a wonderful program because it is government funded, completely free. When you apply to it, they give you money when you're inside the program, and they fund everything you do inside of it. And they provide students with the opportunity to do research with BCM faculty, and you can apply every year. All right. I worked in the Children's Nutrition Research Center. It is a collaborative effort between Baylor College of Medicine, Texas Children's Hospital, and the United States Department of Agriculture. So it's a government building, and I had numerous background checks to go through, and it was a long process to get there. So the kind of research they do here is the faculty do research on nutrition, hence the nutrition part. Okay. For example, they do, how can we combat obesity in children? Or how does your microbiome change with diet? Or microbiome changing from childhood to adulthood? They do all kinds of cool stuff on children there. The project I joined was centered around the problem of premature birth, something I'm sure many of you are aware of. On average, 1 in 10 children are born premature, but these rates are higher in other countries. As you see, the United States is falling behind as well. It's not weird? Okay. The United States is falling behind and medicine because prematurity is skyrocketing here. Yes? Microphone? Hello? Hello? All right. So I'm trying to tell you about the problem of premature birth. I'm sure many of you know about it. Okay. So the United States is bad. Premature babies have many problems linked to their lack of development. One such problem is that when they are fed, they do not grow that well. And part of the research in my lab was over trying to fix how they grow by regulating or changing their diet and their feeding schedules to see the difference between different groups. And I'll get to that soon. Since we can't do experiments on humans, we use pigs to model the human infant. And if you want to know why, I can tell you later, but just know that they're bigger than mice, and they're easier to work with. Here's how the study worked. So we had premature piglets. They were born by C-section early on, before they could be born naturally. And they were split into two groups. One fed continuously, where you just have a constant drip food system where it's injected into them all the time. And the other group was fed a meal every 4 hours, so you just, like a normal meal like we eat. You just feed them a big, large chunk of meal, and you wait the 4 hours and you feed them again. So that's the difference between the two different groups. After about 3 weeks on these schedules, we take their muscle samples, and then it was my job to cut them and look at them using microscopy. So here's some of the work I did. Here you see the muscle was cut as a cross-section. This is what muscles look like, muscle cells. And this is a picture I took. I have many more. I have about 6 gigabytes on my computer. Here's a beautiful image I took at the beginning. Thankfully, I could get this far. Yeah, just marvel at it. And this is a 3D stack that I did of a particular muscle section. And believe it or not, it's kind of useful for looking and determining if the muscles were good enough for growing, I guess, between the two groups. So we analyzed them to see if one group had more muscle growth than the other, essentially. So now I want to speak briefly about the nature of research. If you are a curious individual and you like science and history or whatever you want to do, you may pose a unique question, anything that would interest you. For example, does the color of a barn owl help catch its prey? And you use creativity to find an answer to your unique question. And it's only limited to your imagination, really. So this group in Switzerland put taxidermied owls on a zip line, and they just swung them down to a poor little mouse, and the mouse got scared by the owl. But what they found was that during a full moon, white barn owls caused mice to freeze 5 seconds longer, allowing them to capture the owl. But during a new moon, the darker owls had more chance to capture their prey. So there's this balance between different owls. So that's an example of posing a cool question, something you're interested in, and using creativity to experiment, find a way. There's my animation. Yeah. What I want to stress is that you're not limited in what you do research on. Maybe your questions pertain to medicine, art, history, or engineering. It doesn't matter. And you don't need to know a lot about research either to do it. You can start when you're a child. You can do it, probably. So lastly, I'll just talk about opportunities for you to do research, since now you know that you can. Here at HBU, we do some research here. Some of the students work with their professors. Like a lot of students work with Dr. Chopik. You can go through clubs like Tri Beta. They do biology research, which again, Dr. Chopik and Dr. Ellis over there. Dr. Ellis and Dr. Chopik primarily do and facilitate this research with children. I mean, us. I was stuck in my previous research on children. Okay, research outside of HPU, there's summer programs. You have There's summer programs. You can apply to them. You can find them pretty much anywhere. Here's an example, Pathway to Science. That's a good way to go to find these programs. They're fully funded. You just apply, and if they accept you, you can go. During the summer, you get paid to do it. I don't see why you wouldn't do it. You can go through any fields, mathematics, science, medicine. There's so much opportunities out there. It's kind of sad that people don't do it. National Science Foundation, that's a good one for looking at REUs. And you can ask professors or other students who are more experienced, who they've gone to these programs, and they can give you some advice. You can go through college or university websites to look for programs. Sometimes they have their own special programs that they do, like the SMART program. And that's about all I have to say about outside research. Any questions? I have some special questions for you. No questions? Okay. Preliminary data? No, my job was to... I had no data. Because I had to fix the method so that the postdoc could get the data. But I had really nice pictures, I'm just saying.
Take your little pen.
There you go.
All right. If you can hear me, clap once. If you can hear me, clap twice. If you can hear me, say HBU. Okay. So, kind of at the end here, so I'm racing against time. So I apologize for that. But my name, can everyone hear me? My name is Juan Cerda III. I ignore the title. That's for the project, but it's basically my summer experience. And I worked in the Department of Leukemia under the CPRIT Cure program at MD Anderson Cancer Center, and it's an awesome place. And yeah, but without further ado, I'm going to get right into my research because that's what I focused on. I am pursuing a career in research, going to get my graduate degree, and we'll see where that takes me. But here's, without further ado. So RNA binding proteins, everyone should know what an RNA is. An RNA binding protein is fundamental in several biological processes, and you can see by the little diagram that I made, it focuses on almost every aspect of the cell, from transcription, translation, RNA stability, splicing, signal transduction, and chromatin remodeling. The lab that I worked on specifically worked on HNRPK, which is an RNA binding protein, which stands for heterogeneous nuclear ribonucleoprotein K, which is a family of RNA binding proteins. For today's sake, let's just call it protein K. And basically, the PI that I worked under worked he worked there at MD Anderson, and he just basically branched off from the P53 pathway, and he said, you know what, I'm going to start out my own lab, and let's work on HNRPK. And everyone that works on it is miserable. But HNRPK is so important. Let me go back. HNRPK is so important in several biological processes that when you see a dysregulation in the expression levels, you do see a drastic effect, a drastic change in several of these processes. If it ain't broke, don't fix it. That's how they say. But a lot of the times, HNRPK is dysregulated, and you do see a cascade of effects that might lead to negative effects. And the one we looked at specifically was tumorigenesis. And HNRPK in tumorigenesis is overexpressed in some patients. So we looked at that and we said, okay, let's go forward with what we have. And to do so, but the lab, by the time I got there, they had made a transgenic model in which they increased the expression of HNRPK in the B cells of these mice, and these mice consequently, they were able to produce tumor. They were able to make tumors, and basically we gave these mice cancers. Also, at the same time, we looked at patient samples with diffuse large B-cell lymphoma, which is a hematologic malignancy, a leukemia, And these patient samples showed a correlation with the mice samples, and they also saw that in these diffuse large B-cell lymphomas patients, we did see an increase of HNRPK expression. So taking both of that information, we asked, how does HNRPK behave as an oncogene? What is its mechanism of action that we can go forward to classify this as an oncogene? And to do so, because HNRPK is an RNA binding protein, we did studies to see what RNA transcript it binds to the most, and one of the top hits through the FRIP-seq assay was the MYC transcript. MYC transcript is a proto-oncogenic gene that promotes cell proliferation and cell growth. And we basically just took that information, and here's a picture of basically what HNRPK does to the MYC transcript. If you have increased expression in HNRPK, more of this protein binds to the MYC transcript, which leads to more protein expression, and this MYC protein has negative effects in a sense that it promotes tumorigenesis. So before let's get that down. Let's get this. So stare at it a little longer. Make sure you understand it. The way I put it in my elevator speech, which is a 90-second speech or less of basically summing your entire research, I put it as in a toxic relationship. You know, HNRPK and MYC RNA should clearly not be together, but for some reason they are. So what can we do to disrupt that interaction? So we took everything that we learned. We learned the basic HNRPK function. We took the patient and the mouse samples, and we also took this, the idea of it binding to the MYC transcript, and we asked, what can we use to disrupt that interaction between HNRPK and an RNA binding, an RNA transcript? And that was when the project was basically passed down to me. And we specifically used drugs in these because drugs are they're commonly used for ligand testing. And my project was determining how these compounds disrupt the interaction between these targeted transcripts and HNRPK. And again, here's a little diagram, and I made all these diagrams. They're really pretty. My PI also took them on. He stole them from me, and he's like, hey, I'm going to use these for every talk that I have. So, but yeah, so HNRPK binds to the transcript, MYC, tumorigenesis. But if we introduce a compound, do we stop this can we stop this cascade and eventually stop tumorigenesis? So that's my goal. And we screened over 80,000 drugs, and we came down to about 34 that physically bound to HNRPK. And everyone should know by now that a protein has certain domains that when taken out of its native state, you do see denaturing of the protein. And that's exactly what we're measuring with the assay that I did. So to test my hypothesis, I used the thermal shift assay. In this specific one, we use the thermofluor assay. So here we have our native protein. Am I blocking anyone's view? Okay. Here we have the native protein HNRPK, and then this orange molecule here is the cypro-di-orange. So this cypro-di-orange binds to the hydrophobic regions of an unfolded protein. It's plotted by fluorescence and temperature. So as we increase the temperature, we should see this protein denaturing, and we should see this dye bind to the protein. And when plotted on fluorescence and temperature, the inflection point is the melting temperature. So that's what we're starting with. On the right side, you see when introduced with the compound, does it increase or does it decrease the melting temperature? So that's the assay that we use to basically measure this interaction here. And out of the 34 compounds, I did have 6 that altered the melting temperature. This dotted line right here is significant. So anything above and anything below is significant. Our DMSO control, that's the protein in its native state, is about 43.7. So anything that falls within here would not be significant, but anything above or below would be. So here we have 14, 16, 20, 23, 30, and 25 were the ones that altered its melting temperature by at least 2 degrees. Number 35 was a compound from previous publications, so, and it was also a control. So we that's not a part of the experiment. But in summary, HNRPK is an oncogene when expressed and is sufficient enough to drive hematologic malignancies. So therefore, because we don't know that much about HNRPK, therapeutic agents are needed to stop this HNRPK's oncogenic function. And as far as my project, 6 of the compounds that we tested had a plus minus of 2 degree shift, and one of them, number 25, increased while the rest of them decreased. And I'd like to acknowledge, everyone from the CPREP program, my mentor, Sean Post, and the genetics department, and the grants that they're under, and also the CPREP grant that gave me the opportunity to come here. And here's the building that I worked in. Here's Sean, my PI, the research tech, MD, PhD student, my mentor, Prerna, me. There's Sean again, and me. I can't say enough about these people because they were awesome to me. It's a small lab. A lot of people went to large labs, but I got the privilege to work hands-on with my PI. He was doing bench work with me, and he was really awesome. So I'm really thankful for them. Also, this was the first thing I did on my own, which was a maxi prep. And then there's me presenting my poster at the symposium. There's me at the poster, and I also got to see zebrafish. So I like fish, and I said, hey, I'm gonna be working on zebrafish research later in the semester, and my mentor Prerna connected me with Oscar, who provides us with the embryos, and I got to see a lot of cool stuff that they're doing, and just the network and the community that these people have is so awesome to to deal with. And I'd like to acknowledge the HBU school and COSI for allowing me the opportunity to be here in the first place. And I want a quick thing why I chose MD Anderson. So first of all, it's in Houston. This is my city. I love it. I don't imagine living anywhere else, but I love Houston, and there's so much in this city that I want to give back to, so I chose here. I also want to work with cancer, and MD Anderson is the #1 cancer center in America. I also have a best friend that's inpatient at MD Anderson right now. We've been friends for quite some time, since 6th grade, and he's fighting AML right now, and he is inpatient. They don't have a cure for him as of now, but I would have included him, but out of just respect, I decided not to. But I do want to talk to him because he really did push me to do this. He's like, hey, and I, really coincidentally, I studied my internship on his birthday, and that moved his mother to tears. And I actually got a call one time when I was at work, and he's like, hey, I'm sick. I'm gonna be moved to the ER. And I said, hey, Sean, can I take the day? Let me go. So, and it was really funny because we were meeting up as, he's coming down in the bed, and he's a really funny guy. He's cool, but just knowing that there's someone out there that needs that help is what's driving me to, to pursue a career in research, and specifically in cancer. I also want to thank the CPRIT program, CPRIT Cure. Dr. S., who was a faculty here, also was a great, program director, and I encourage everyone that goes to HBU that you have a great chance of getting to these programs if you know people and you talk to them. I also want to thank the professors. I think was it Dr. Horn once said, what you learn once comes to the light again. And it's crazy because the things that you learn in your class come back later. What you learn in genetics, endocrinology. I'm learning a lot about immunology that's making, stuff click right now. And in micro as well, general physiology. It's crazy how everything that you learn eventually comes to the light. You just hope that you know it and you recall it and not just memorize it, So, and I want to thank some friends specifically, Jacob and John, who I don't see if he's not here. I sat up there, and when they were giving their Under the Microscope talk, and I said, I'm going to do this. I said, I'm going to get with these people. I'm going to ask how they did it because that was interesting to me. And people left and right of me were snoring and stuff, but that's no big deal because that interested me. And I really want to thank Jacob because, right after he we sat down and he gave me a piece of paper. He said, these are the programs that you need to apply to. These are the things that you need to look out for, this and that. And I clearly remember, like clear as day, he came up to my face, he looked around, he's like, you need to apply to these programs now. So I'm like, okay. So I really want to thank them. I also want to thank my family for being so awesome. My dad and my mom are right there. You know, I really want to thank them for pushing me. And my dad said, you know, don't worry about tuition, don't worry about anything else. I got it. You just do what you got to do and come out on top. I want to leave you with 3 little bits, and I'll be ending. Never give up, please. You know, there's often times where you go through rough times, whether it be in school, whether it be in family. Don't give up. You have hope here with your friends, with your family, with the professors. I also want to say to be passionate. No matter what it is, whether it's music, sports, helping others, arts, be passionate and stick to it. And lastly, make the best of your opportunity. And I actually didn't know Oscar was going to put his slide up, but basically, you know, thank your blessings, you know, to whoever you see above you. To me, it's the man upstairs, and I want to give all the thanks and everything to him. But I also want to say, never give up, be passionate, and make best of the opportunities that are given to you, because you don't know when they're going to come around again. And just make best of everything that you have. And yeah, that's it.